Sí, pero también más claro. Es que ya no me conviene ponerme, creo que están grabando. A ver, video no estamos mal, nada más grabando. Ah, te digo.
Dr. Servien earned his PhD in Biomedical Sciences with honors from the Universidad Nacional Autónoma de México, where he currently serves as a professor at Inés Morelia and co-founder of the Microbiomics Laboratory. His research focuses on the genetic ecology of microbial communities, exploring microorganisms and viruses from extreme Mexican environments through genomics and metagenomics. During his academic training, he conducted research at the University of Copenhagen and completed a research stay at the Baylor College of Medicine. He is a member of Mexico's National System of Researchers and has received the National University Distinction Award for Young Academics at UNAM. Internationally, he has served in leadership roles within the American Society for Microbiology and currently acts as an early career ambassador in Mexico for the International Society for Microbial Ecology. Please give a round of applause to Dr. Dominic.
So, hello, good afternoon. Thank you for being with me. I'm a storyteller, so I'm going to do my best to provide it with some stories about the work that we are doing here in different places in Mexico with my students. I'm from Morelia. I hear some people in here is from Morelia. So I'm from Morelia. I study at the National University of Mexico in Cuernavaca. And then I moved to Mexico City. And then, surprisingly, I came back to Morelia to start working. Actually, 2 weeks ago, I just start my 10th year working at the campus here. This is the UNAM campus in Morelia. So it's going to be some stories about the work we are doing. The National University of Mexico, since about 10 to 20 years, started creating new campus around the country. And this was wonderful for me. Don't worry. Don't worry.
The sound is not getting through.
Oh, okay. Well, I never imagined I would be fortunate with biology, to be honest. I really like to draw, to travel. But then I discovered under my bachelor that I was good in biology. So I decided to start my journey at UNAM studying genomic sciences. At that time, it was the second generation of students working with genomic sciences. I guess it was the first program in Latin America working with genomes and informatics. So it was very interesting to me because my parent was a computer teacher, and my mother studied chemistry. So I was the black home working with science. This is my lab. I share this lab with another researcher, that is currently the director of my university. So here we work with microbes. We decided to create this lab to provide an opportunity to students that are interested in working with microbes in the western part of Mexico. So in order to they avoid traveling to other places like Mexico City or whatever. So this is the focus of my lab for my group. We study viruses and extremophiles. It's my main area of research. Viruses and extremophiles. But we also started working with plants, animals, and other microbes that are associated with animals and plants. Basically, my students came to me and said, Luis, I love tapirs. I love this plant. Can we study the microbiome? And I said, yes, because I'm not good at saying no. But the other love that I have is for monarch butterflies. I'm sorry, my throat is not so good at this moment. Because you know, the monarch butterfly is the national insect for Canada and the United States, and here in Mexico is a very emblematic insect. So at the time, nobody knew about the diversity of microbes that resides within this insect. And we also work with education. We focus some of our research about how to teach students, mainly bachelor students, about science. What are the best techniques or programs or simulations or laboratories or whatever to better teach and stimulate the students about microbial research. So I love to travel. I save every penny since I was 17 years old. My family cannot afford traveling, so that was like a mission too. It was like a passion for me. So before I started traveling to other countries, I was very focused on traveling around Mexico. So I guess about 5 years ago, I was able to visit all the capital cities, states, and I only have like 30 magic towns in the list to move around. So for me, that's an inspiration. So the first big hope message for you is me to be passionate about the things you like to do. So for me, traveling is like an important activity for my mental health as a teacher and as a researcher. For me, it's very important. It's like a stimuli. So maybe for you, it's the sports, science, culture, I don't know, drawing or something. So don't ever stop passion about the things you like, because it's going to be very important for the future. So the main topics for today is about speaking about microbiology and some techniques about genomics. I am not sure if you are accustomed about these topics. I don't know. Can you raise your hand if you ever hear genomics? Okay, there are some hands. Great. So, and for the second part, we're going to start talking about extreme environments and Science University focuses on some of the topics we're working on the lab. So the first thing that is very important for me to tell you is that we are a microbial world. We live in a microbial world. I don't know, can you put your hands in front of you? So that then, what can you do? That's the things we are able to see. But there are more. There are a lot of microbes in your hands. Actually, if you wash your hands with the best technique, you know, in about 15 seconds, you're going to be covered in microbes again. So I hope you are not scared about microbes, but they are good. They are friends. They are always with us. Since the moment we born, we all are always covered with microbes. Actually, the first microbes that we receive are from our mother's bodies. It's very important. It's the only time in our life that we are going to be able to receive these microbes from our mothers. So I don't know if you ever hear this, but it's called the human microbiome. All of us, this moment, I hope all of you are healthy. But at this moment, about 200 grams of your weight are microbes, like bacteria, I don't know, worms, viruses, and we are healthy. So microbes are our friends. So we have like another organ inside of us, so that microbes. And we don't know, well, I'm assuming you're not knowing the names of these microbes and what are they doing in our bodies. So that's the main topic I want to start with. So we have this number, it's like 38 trillion cells that are not human. They are bacteria. And we have 30 trillion human cells. So we are, in some sense, more bacterial than human. Okay? And we're healthy. So microbes are always with us, and this is excellent because we just passed the February 14th. So don't ever cry about your girlfriend, your boyfriend, or whatever. You are always surrounded by microbes. They are going to be with you until the day you die, and they even are going to be growing in your dead body. So, for them, you are their fun. So whatever you do with your body, the things you eat, the people you meet, the places you visit, you are going to be always surrounded by microbes. So if you're sad, they are going to be sad. If you're happy, they're going to be happy. If you eat healthy, they're going to be okay. But if you don't exercise or you don't do something, they are going to be in a bad state. That is called viruses. So in some sense, we work as a holobiote. That's the concept. That's the term. We are, in some sense, human, but we also have bacterial, archaeal, and viral, and other eukaryotic microorganisms inside of us. And we need them in order to function properly. So the name of that conjunction is holobites. It's like an holistic term to refer to a plant with the microbes in the roots, in your skin, in the ribs, or in our body, and the microbes that we have from the hair to the fingers. We are always surrounded with microbes, and we are working together. So this is, an important topic. So several scientific journals constantly cover this research. This is like a nice cover from Nature that is called the Other Genome. It was several years ago. But we have 100 and more unique genes than our own human genome. So we have more bacterial genes than human genes. Human faces contain about 1 million bacteria per gram. So I like to joke about this number because every time you go to the toilet and do some poo, you're missing a lot of bacterial cells and you are more human. So remember that. Every time you go to the toilet, you become more human. And this is important. We have a lot of bacteria in our intestines, mainly in the final part. And most of them don't like oxygen. It's toxic for them. So in some sense, you can look at the mirror and look at yourself, and this is the human part, but also all the microbial part. It's fascinating. So if you don't know the names, it is like a nice resume. We have bacteria, fungi, viruses, and archaea all the time. And we are healthy. So the thing is that the first time we hear about bacteria is when our parents take you to the MD because you are sick, you're feeling sick, and the doctor told you, oh, you have a bacterial infection, you need antibiotics. That's for most of us, the first contact with microbes in the body. But we have a lot of other stuff. Actually, there are more viral particles in our body than bacterial cells and human cells. But that's another step. And we have this name here. Have you ever heard? I'm curious. Did you ever hear this word? Yeah. Yeah. Oh, nice. Great. Some of us do. So most of these archaea produce nitrogen. Decimal gases. So you are going to remember. By the way, there are some bacterial species that can cause disease. You know that. Also, viral particles that can produce diseases. But until today, there is no archaeal species that produces a human disease. So they are friends. You know, this is the digestive tract. There are some scientific names for some microbes, but you know, the final track of the digestive system is by no means the best place to find microbes. Okay? These are quite important actually, because they are helping us to digest the foods we eat. For example, when the human genome was sequenced to the first time, people was very happy because they believed they would find all the genes related to digestion, but they could not find it. So how can we ingest the fruit in the morning or the vegetables? Oh, it's because we are not doing it. Microbes are doing it. The microbes that reside inside our bodies. By the way, some of these microbes are working in this axis, the good brain axis, which is the hot topic in the field. Because we have a nerve, that is the vagus nerve, you will remember forever, el nervio vago. It's the only nerve that connects the intestines to the brain. So I hope you all eat something, but if you don't, please be patient with me. If you feel this like the sensation, like I'm not feeling good, I'm going to sleep, I really need food, I'm tired, it's because microbes are telling your intestines that food is needed urgently. And they sense this and provide the information to the brain. So, for example, when you have some eating disorder, or when you feel sad, constantly sad, or you have like learning problems, there have been several studies that relate microbes, these proportional microbes in the intestines, with some of these diseases or phenotypes. So, in some sense, you're asking like, I want to eat this apple because I want, or is the microbes? So let's say like, I need the starch, I need sugar, I need blood, and I interpret like I need this apple. So is the human part, or is the microbial part that is telling our bodies what to eat? So remember this name, the gut-brain axis. It's all the signals, molecules, hormones, and everything that is being sent by the vagus nerve directly to the brain. And this is important. I don't know if you're familiar with some of the neurotransmitters, but one of them is called serotonin. Is one of the main neurotransmitters related to happiness. So this neurotransmitter is not produced mainly in the brain. It's in the intestines. It's like we have a second brain. Okay. So when you hear about neurotransmitters, I'm sure you always think in the brain, but that's not true. For several neurotransmitters, most of them are produced in the gut. So I don't know if you are related to this. I eat, I feel happy, I produce serotonin, my microbes are happy, I'm happy. But if I don't eat, and I'm listening to Luis at the, 4 o'clock, and I'm feeling hungry and tired, probably microbes are feeling not good. So
I have a question.
Sure.
Be patient with me because I'm not a doctor and I don't know anything about chemistry.
Okay.
But there are some meds that actually delay the absorption of serotonin, and that is only at the brain level, I'm assuming. I don't know. But how do those, for example, affect all of this idea that we also have serotonin in the bowels and everything?
Yeah, well, actually, some of these meds act not only on the brain, they actually work in the intestines. I'm not an specialist,
Me
but
neither.
there are some people that feel with gastrical pain all the time.
Okay.
The only solution is antibiotics. So that's not good, because antibiotics kill like antibiotics.
Yeah.
So if we rely on antibiotics all the time, they are going to kill several types of bacteria. Some of them are friends. Only a few are the enemies, and not all the time. So these meds are controversial because I don't know if you ever feel sick and the doctor told you to take this antibiotic. You recover, but the first day you feel sad and tired. That's because you're killing your microbes, you're killing your friends. So you are not digesting well, you're not feeling good. So yeah, they have an effect on receptors. Just to be more specific, there are several receptors that constantly communicate with bacteria. You know, the living bacteria, but also their metabolites or their metabolism products. And that they are received in the intestinal tract, mainly in the epithelial cells. That's one part of the response. The other one is because they train our immune system. Yeah. So, this is good. No. It's good. No, it's fine. This is actually quite important because one message for you is that when we see this, your baby is dirty. So the first thing is we need to clean him or her. But that's not the best option most of the time because we need to train our immune systems since the first years of life. That's quite important because otherwise we are not able to train our immune system with microbes, and they are going to be very responsible for allergies, for example, or intolerance for some food. And that's because babies were always clean, and that's not good at all. Well, other names. I put some black letters in the sizes for you to try to remember them or future life maybe. The main bacterial groups that we have in our body, mainly our intestines, are called Bacillota and Bacteroidota. All of us have it. They are very common, they are very abundant, and most of them are friends. And this is a bacterial genus name. It's Bacteroides. This is the main bacterial genus that we have in our body. We all have them, but the thing is, it's very variable. So we all have it, but it's very variable between us. So, you know, there are several research papers telling you if you have this Mediterranean diet, you're going to have more of these bacteria than the other, but no, it's not always the case. It's only correlations. It's not outside. That's very important. And we also have microbes in several other places of our body. In the nasal cavity, oral, skin, gastrointestinal, and urogenic. This is very important for health. You can think about this part of the body like a dessert. It's dry, it's hairy. But if you think about the armpits, it's always wet. It's like a jungle, like a tropical jungle. So you can start working with ecology, with the interactions between microbes in our own body. So you don't need to go to the tropical jungle in Brazil or here in Mexico. You can work with microbes in your own body. So this is a reason for this part. These microbes help with digestion. They protect against pathogens. They provide nutritional nutrition. They're important for nutrition. They train our immune system. And when we have some disturbance in these communities, because these are communities, in the skin, in the lungs, in the brain, in the liver, and in other parts when proportions of microbes are not in the best shape, probably you have, and that's why you're cyst, and that's most of the time related to disease. So, am I feeling sad because actually I'm sad? Or is it because my microbes in my digestive system are not in the best proportions and they are not working in the best way? And this is related with a lot of stuff. Dementia, Parkinson, Alzheimer, a lot of stuff that is critical in our current time. Also, with cancer. Here in Mexico, I guess we are going to be about 2 to 3 of us that probably are going to die because of cancer. And there are some microbes that are associated with the cancer tissue. Are they friends or are they enemies? Microbes can produce cancer, or is cancer recruiting microbes? This is a hot topic of research. There is no definitive cure. But the thing we do know is that there are some bacteria that actually enters inside the cancer and recruits white blood cells that actually are good at killing cancer cells. So this is important for therapeutics, for medicine. This is a nice example. Mice can be shy or they can be brave. And you can do, actually, this is a nice laboratory test if they are afraid of cats. So you can put a cat in a box with mice and you can very fast know who are brave and who are shy. If you feed these brave mice with pellets, with food, that have been inoculated with microbes from the faces from these mice is now shy. If this shy mice eats the pellets with the microbes from these mice, it actually becomes productive. In Drosophila, the fruit flies in the garbage, the sex preference is related with microbes. The female attraction to males is related with the microbes in the bowels. You are fruit fly from the same bloodstock, and the only difference is the microbes you have in your body. That's the difference between mating or not mating. Oh, because the hormones and metabolites that these microbes produce. And I hope at this time you always remember that we are surrounding microbes, but they are not the same always in your body. When we born, every time we try a new food or a new drink or mother's milk, we are in contact with new microbes. So imagine every time you eat something, you are discovering a new world of microbes. So that trains the immune system. But when you are a teenager or a young adult, that is actually your age, is the moment where microbes are in the most balanced state. They are in equilibrium. Because you start traveling, because probably you have your first sex relationships, or you have your first travels abroad. I don't know, you're in contact with people, with society. And that's the base state that we call that is in equilibrium in your life. When we get old, our communities are sadly very imbalanced. They're unbalanced. They receive a lot of microbes, some microbes that at some point in their life were friends, now they are potential enemies. So that's why some people at the late 60s, 70s, 80s, they start having these infections constantly. And that's because our immune system is not working properly at that age. So we cannot have these communities in check. So this is dynamic, this is ecology. This is dynamics, ecosystems living inside our body that are changing through our life. And this is just a comic. This is a timeline with the main geological moments in the history of Earth. Here you have the formation of Earth about 4.8 plus minus million years ago. I'm sorry. And from here to here, the life on Earth was only microbial. Other animals, flowering plants, land plants, fish, are very recent. So for the most part of the history of this planet, life on Earth is microbial. So this is another important thing to take home. For life is a microbial world. It's not a human world. It's not a plant or animal world. It's a world dominated by microbes. So, is this relevant or not? Well, the thing is that we currently, as a human society, an intelligent human society, face several problems. And when we look back at biology, we discovered that many of these microbes have the solution to many of those problems. That's called bioprospection. So, please remember this. For about half of the life of Earth, it was microbial only. So if you look at microbes in the atmosphere, on soils, on caves, several kilometers beneath the surface, you always will find microbes. They are all around the planet. In the air we breathe, when we take a shower, there is always a microbe. So I don't know if you ever hear the 5 kingdom classification of life. This was proposed by Robert Whittaker in the 70s. And it's very didactical. You know, you have animals, you have your dog, your cat. You remember that you see something green, it's a plant. There is some things that look like plants that are not green, and you say, okay, these are fungi. Later on, you remember that there are some cells that have a nuclei and other some are eukaryotes and other prokaryotes. So this proposal is from the 70s. And well, I don't know if you ever have this on your classes, but in several places in Mexico, it is still being teaching. Okay? But it's nice, you remember, you know, an animal, a plant, a fungi, some microbes, some with nuclei and some others don't have it. It's easy to remember, you know, it's familiar. But have you ever wondered why we have fly? I was curious, so I read the original paper. It's a science, it's a nice paper. It's hard to follow, but the main difference between these 3 kingdoms, plants, fungi, and animals, is the way they find food. Believe it or not. I was kind of skeptical when I read it in the paper, but you know, plants do photosynthesis, fungi do absorption, and animals do ingest food. But you know, nowadays we have exceptions for all of this. So there are some plants that are not green. Actually, some fungi hunt for food, and some animals rely on microbes for nutrients. So believe it or not, for several decades, the way we teach biology is with 5 kingdoms until today. And the main difference between these 5 is the way they obtain food. So it's not big science. But at the same time, some researchers like Harwood and his colleagues start working with molecular biology. Actually, the first paper was published in the 70s, actually in the same year. But they don't focus on names. They focus on data, on molecular data, to try to figure it out how to call stuff, not like phenotypes, but with molecular biology, with molecular sequences. using marker genes, using DNA. At that time, sequencing a single gene was like a PhD thesis with honors. And actually, it can be kind of dangerous. So they actually obtain DNA from some animals, from plants, from bacteria, and obtain a marker gene, the DNA sequences from the 16S somalia gene. I don't know if you're familiar with that. It's a gene that is related with the ribosome, that is related to the production of proteins. So we all have it, but we have a small difference in the DNA sequence, so we can look for that difference and try to create an phylogenetic hypothesis about the relationships using DNA. And they start doing it with less than 20 sequences. And surprise, you don't have 5, you have 3 main groups. And they are called domains. And that's the way we should teach this to new kids. So we have 3 domains today. Until today, here's the 3. Bacteria, archaea, and eukarya. So all life on Earth is between these 3 domains using molecular biology. And you will ask about viruses. Well, the thing is viruses, their genomes are very, very small. They are very different, and they don't have the same marker genes that we use to create the life in a genetic tree. So they are screwed because they don't have ribosomes. So they don't have the ribosomal gene that is used to create these hypotheses. So that's why you never find like a branching here that is biased. That's why. So this is a nice paper in Nature Microbiology from a few years ago, and the name is fantastic. A new view of the tree of life. So this is the same idea to create an hypothesis, like a phylogenetic hypothesis, but nowadays we do it with a large amount of data. And the 3 domains are still true. Here you have bacteria, this is archaea, and this is eukarya. But the vision is different. It's not the same as this. You see, bacteria is early branching, and archaea is more closely related to eukarya. But look at these 3. Archaea are very close to eukarya. Actually, in some hypotheses, in some phylogenetic hypotheses, all the eukaryotic domain is inside the archaea domain. So archaea are very close to us in the history of life. Actually, just a few years ago, a Japanese group identified a group of archaea that is called Archaea. Archaea is the name. And they contain genes that a few years ago were only eukarya minority. So archaea were very important in the transition to complex life. And bacteria actually has many more branches. And I don't know if you can distinguish this like purple color. I don't know if you at the very back distinguish this purple, but all the purple branches are microbes that cannot be cultured. So we are not able to have it in the lab, like like an petri dish. Most of the microbes are unculturable because we don't know how to grow them. That's the state of the art. So you cannot do, the classical microbial analysis with culture. So you have to rely on molecular sequences. Even if you don't like it, it's the only solution for several microbes. And this is every single line. I don't know if you're able to look at this. Every line is just a single, how do you say? A single, animal or plant that covers an entire group of organisms. It's
like a representative of the.
Yeah, it's like a single representative for a bunch of organisms. So this is the strength of biological diversity. And this is amazing because we humans, belong to a group that is here. It says of histocontact is this line with all the other animals. and close relatives to animals. It's this single line. And all green plants are here. So when you say, oh, life is good, we have animals and plants, you're referring only to this line and to this line. And you have all of that. So that's why I discovered that I needed to work with microbes. Please just assume. Right. If you ever hear that you are an epistocon or an ovazoan, you're right. That's the taxonomical specific names to refer to animals. And if you ever hear plantae or archaeoplastidae, that refers to plants. And green algae, red algae, and golden algae. So this is amazing. We have all this information available, it's public, in a database that is called GenBank. Have you ever heard about this database? Okay, the first time I hear about this, I was like, whoa, I have all this data in my computer, doing everything I want and do everything with my computer. So that's called bioinformatics, when you can analyze electrical data with the help of computation and informatics. So if you never heard about this, just Google it, GenBank, and you will find the link. If you have it, you can take pictures, I don't know. But we biologists believe that we produce a lot of data. That's actually not true. The main group of data, like in large quantities, is coming from astronomy, from physics. Second place is biology. So we have a lot of data. We have a lot of geological sequences available in databases. And actually, now we live in the biology of movement, I will say like that. Because this is a sequencer now. It's your error. We work with huge machines that are called sequences. So nowadays you have these small things that are called Oxford Nanopore Mini Ion. Actually, they are cheap now, about 20,000 pesos, I guess. It's not that expensive.
The import has been a nightmare.
Yes, yeah, and the rabbits and
Yes.
yeah, I know. And here you have the first woman scientist that actually was not a biologist, working with genomics in the International Space Station. So this picture is from space, it was taken in the ESS. Here you have the sequencer. You know, because there is no gravity, everything has to be blue. So this is the screen. I don't know if you're able to look, but there are some bars in here that are representing the on state, on real time sequencing in space, the first time. So nowadays we live in the mobile genomics era, and these are the three main technologies for sequencing. Illumina, PacBio, and Nanopore. And these are all numbers from last year about the number of genomes, the sequence read archive, and nucleotides available for each domain of life. So we have a lot of bacterial genomes, but we don't have a lot of eukaryotic genomes. So even if it is easy, some groups of forms of life require more genomes. So you have all available this in your lab and your cell phone and everything. So when I was a student, your age, the thing, it was completely different. Every single sequencing run cost about $500,000 pesos. My PhD students involved only 2 data sets that cost $1 million pesos. And nowadays you can sequence some of these just for 20,000 pesos, for example. So completely different moments, completely different economies. Everything was different, just between your age and mine. And it's very cool, not so many years. So this is just another way of looking at the same tree. You have bacteria and archaea, and we are this single line. My ecology students told me that I am joking with this picture because I dismiss carrots, but actually that's not true. Another thing is that all these groups that you can see here are special groups of bacteria and special groups of archaea that don't have the ability to metabolize many stuff. Some of these bacteria are not able to synthesize their own DNA because they lack the genes necessary to create DNA, and they are alive. So most of these bacterial and archaeal cells are very small, and they lack essential genes. That's part of the cause. Most of these live on caves, on the underground. And they depend on interactions with other bacteria or archaea for their metabolism. So this is just another picture of eukaryotes. This is a phylogenetic tree only for eukaryotes. And we are here in Ovazoa, in the group of Amorphia. So these names are very hard to follow. You don't need to memorize. But it's just to represent the huge diversity of life, even between or close neighbors. And nowadays you can produce this type of analysis. This is called phylogenomics. You use genomic data to relate to phylogenetics. And you can use your laptop, any of those laptops you have. In just 30 minutes, you can create this image. It's quite easy. It's quite easy, actually. So what do we do? With examples from animals, plants, soil, whatever. You know there are microbes, and you extract and purify the DNA from the whole community. And then you sequence it using the technology you like or the one that is affordable for you. And that's called metagenomics. You are sequencing the DNA from a community, not from a single microbe, from a whole community. And in that way, you will have data to answer these questions. What microbes are present in my sample? What are they capable of doing? How they do it? And are they going to be helpful for some process, like, I don't know, biotechnology? So you can do this, the prospection. So you look at microbial communities to find solutions to human problems. So these are important terms. Microbiota, all the set of microorganisms that reside in a sample. You say the human microbiota. I have A, B, C. Microbiome, the set of microbial genes or genomes present in a sample. So now I'm referring to the set of genes or the set of genomes present in a sample. All the same. So microbiota and microbiome is not the same thing. They are related, but it's not the same thing, just for vocabulary. And this is a problem. More than 50% of the genes in the human gut have not been characterized. So we don't know what they are doing. So we know we have microbes, but we don't know what they are doing. That's the state of the art. And they live inside us. And they want to live with us all this. They have been with us all this time, all these years. And they are going to be forever with us. So the problem is bigger because in some samples, about 99% of the microbes in there are not known how to filter them. So that's why I told you omics is important, because even if they are not, we don't know how to culture them, we can start DNA. So we can look at DNA to see what genes are present, and maybe we can have some clues about the function in the ecosystem, in the soil, in the body, in the air, in the water, whatever. So this is genomics when you have a single bacterial or archaea or viruses or plant or animal, your dog, your cat, whatever, and you extract DNA with the intention to recover, like with computers, the complete genome to ask that type of questions I told you about. And with the metagenome, you have the ability to recover many populations with many cells. Each cell represents a single genome. Would you mix it together, like in a shaker, and extract DNA the best way you can, and then you sequence the best way you can, and then you try to recover the pieces that are very similar and try to say, okay, these pieces are from this population, these pieces are from another population, to try to recover genomes from communities. Okay? So this is big data, I can already told you about. These are some of the main genomic techniques, genomics, that's dealing with DNA, transcriptomics that relates with RNA. So when you look at the genome, you see the genes that are present. This genome has the potential metabolism for doing whatever, but you don't know if it actually is expressed or not, because DNA stores information. But if you want to see if it is expressed, if these genes are expressed, you rely on transcriptomics. If you want to look at proteins, the main actors, the chemistry, enzymes, I don't know, cofactors, for example, you need to look at the proteome. And there are a lot of other omic techniques, metabolomics, dealing with sugar, nucleotide, amino acid. The main idea is that if you have more, when you increase the amount of information, in theory, at some point, you will be able to identify potential phenotypes and functions. And it's a really nice term that I really want to tell you about, that it's systems biology. For the ones that I take in the classroom that actually really love biology and computers, future area of research with a lot of potential is called systems biology. So systems biology is actually an holistic term that refers to seek to analyze interactions among different molecular entities to provide holistic view of biological processes. So in this picture, you have networks. This is the best way to look at this amount of data with networks. Ecological networks, I don't know. I guess you use TikTok or Instagram or X, something, I guess. So I can ask you, like, can you borrow me your cell phone to see what type of content you are looking at? But that's going to be for a single person. So if we are the subject group, we can create a statistical map about the content you look as a group. So probably some emerging properties are going to be now visible with the increasing amount of data. So if I have more data, there are going to be visible new patterns. Like at what time I need to do these social networks, what kind of content you are looking at. So I don't know if you imagine the possibilities of this. But I'm here, we're talking about biology. So the more you go in this direction, the more complexity you have. So here with the genome, you have, genetic regulation and genetics. I don't know if you ever heard these terms, but you're dealing with genomic data. If you have transcriptome, proteome, and metabolome, you are having more data available that is more complex in article. So you cannot look at them in separate. You need to look at them together. And the best way to do it is using networks. So I don't know if you ever have a math class about networks and coefficients, correlations, or something like that. Well, it's important if you relate to these things. The best way to look at this data is with networks. It's the future. So probably you're accustomed to this type of projects. Biology produces insights. This generate new biological questions, so we need new technology. And with new technology, we have new data, formats of data, and this related computation. So we need new computers, new software, new tools, and then this open the field for new hypotheses. So it's like a circle. So I don't know if you like social sciences, arts, biology, or whatever. We are all together, we are all related, and we are all needed. Most of the major fields in social life currently is several topics at the same time. So in the few minutes I have, I just want to tell you about some examples from the research we're doing. So this is a place called Los Azufres in Michoacán. It's a geothermal area. This particular hot spring is about 98 Celsius degrees, and the pH is 2. So this is the whole people that is working with me. We have been to this place several times, take several samples. I'm going to be very fast. So we have another hot spring, and we were able to sequence the Pierce archaeal genome in Mexico with Mexican science. with Mexican students using Mexican technology. And it's called Sulfolopus acidocardiaris. So if you wonder about an archaeal cell, this is an archaea. Say hello to that. We sequence the genome. This is a map of its genome. It has this amount of genes, easy content proteins, whatever about this genome. But we were also the first to sequence viruses. Here, just about, I don't know, like 6 hours from here, like driving to Los Azufres. The surprise was that we found new viral particles. So people go to this place and swim in this place and put mud on the skin. It's important for public health. So these are the particles here in Mexico. They have these shapes, and this is their genomes. Most of the genes are unknown function. So we need to go to very far away. We cannot wait for the next pandemic, because just a few hours driving here in Mexico, we can find new viral particles totally new to science. These are other particles that are lemon shaped. I don't know, I cannot see the lemon part of the name, but maybe these ones. They are lemon shaped, they have their genome inside, they have circular genomes, they have a conserved area and a conserved area, and again, most of the genes are of unknown function. And we have it here in Mexico. There are many opportunities to work with that, because these particles are there. We have the pictures. These are viruses in the environment. But actually, we don't have the insights about it. We only have pictures. We don't know how they are called, if they are problematic or not. We're not sure. We only have pictures and some sequences, but they are so new that the genomes are not contained in databases. So, are they friends? I really hope. But that's the same. We don't have, actually, we have a lot of places here in Mexico that have not been analyzed for the microbial or viral diversity. So just imagine the possibilities in this huge country. Here, believe me or not, there is some green microbial communities. There are some archaea that are heart-shaped. They're very good. And they have interactions with viruses and with other archaea. We can sequence the metagenome and identify genes and try to create hypotheses about the metabolism, biochemistry. Here is the Krebs cycle, glycolysis, probably you have a class about that. So we look for the genes that produce those enzymes and proteins in genomes to try to figure it out what are they doing. We identify a lot of new bacterial genera using molecular biology. We were able to sequence some algal cells. Believe me or not, this is an algae. It's very small, actually. They are actually the eukaryotic genomes with the smallest genomes available. It's a new species, and a new genome currently lacking a name. We only have sequence, so that's the way you see cyanidase SP, but because it doesn't have a name and a last name. You can grow them in the lab, and you can work with ecology. These black spots are fungi, the green part is algae. I don't know if you can see the yellow arrows. These are bacteria that some of them produce something that is killing algae and fungi. So they are inhibiting the growth of these other community members. So there is potential there. So we sequence genomes and everything. We also do the same with some colleagues from Chiapas, working with the crater lake of the Chichen volcano, doing the same, trying to figure out the metabolism, the microbial metabolism in the crater. Cristal from Uruguay, she is my PhD student currently. She is working with the Sonora desert. She and I visit Sonora. It's quite an adventure, I would say. She's looking at the microbes. It's actually from here. This is the picture from the study site. So she's taking samples from the surface of these sand dunes to look for microbial life. We have a lot of deserts. We have a lot of them. We actually start working during pandemics. So she said her PhD during pandemic, so we were unable to travel the first years. So we moved to another place here well, there in Michoacan, that is called Ciporani Fiernillo. It's just in the way to Ixtapa. Probably you been there, and you will see the how do you say, the CP electrical company. So just in front of that, you will see some of these type of soils, and we look for the microbes in there. And surprisingly, we've found a lot of bacterial genera that are known to be extreme, like extreme. For example, the Inococcus. The Inococcus is able to live inside the nuclear reactors. So most of these microbes are able to survive on the surface because you have radiation. Just look here outside, the amount of solar radiation we have. So it's hard. So imagine you're a bacteria in the soil. You have to be able to resist to those conditions. So all of this microbial life is able to live in there, under about almost 52 Celsius degrees with no water around. So she's working with that. And this is the place we work. This is the place we love. This is here in Mexico, it's called Sonora. This is not Arabia, this is not Africa, this is Mexico. And just in the very few moments, just to tell you this story, because currently we're having the monarch migration. So all monarch butterflies have microbes in their gut. And these are the names of them. Some can be cultured, some others we don't know how to culture them. They have beta protobacteria, gamma protobacteria, kinobacteria, protobacteria, and firmicutes. But the main bacteria that monarch butterflies have on their guts is called Commensalibacter. So we were able to culture it. It was a lot of work during my PhD. The secret was to put tomato juice from Walmart. So this is the bacterial cells. This is a intestine from a monarch butterfly, and the red spots are red crew to detect the DNA from the bacteria in the tissue. So we sequenced the genome and the metagenome, and we were able to recover the genome from this bacteria. We published it and we proposed a new species because we performed some comparative genomic analysis, and we obtained several amount of information telling us that it relates to a new species, and we call it Commensalibacter papalotum, because you know papalotum from the Nahuatl word means butterfly. So it's the bacteria, the commensal bacteria from butterflies. And we performed some phylogenetics from the commensal bacteria, from several nectar-feeding insects. So we have the monarch ones, the Drosophila ones, from bees, and from a group of Phyllocopa, bumblebee. So each of these insects have a bacterial component that is important for digestion because these are acetic acid bacteria. So when you smell vinegar, that the smell is because acetic acid. So these insects rely on energy for migration because of the bacterial metabolism. During the winter, they don't feed, so they store fat in there. If you ever happen to drive in a place with mosquitoes or something, you see the window, it's like a yellow spot, that's fat. So that's the way insects store their nutrients. But during February, March, particularly monarch butterfly, the metabolism is awake. So they need to metabolize all that fat, and it's very important for migration. And here, this is actually a really nice, this is the last. These are stick insects from Crete. So I stayed at a friend's house, and he has a garden, and I was looking around the plants, and I found these stick insects that are huge. So there are some people working with insects, and they told us, "Oh, this is a new species." Like, seriously, in Crete, in the urban area of Crete, we have new species of stick insects. Well, that's true. And one student, Hector, performed her ecology pre-grade working with these insects and their microbes. This is the gut of an stick insect. It's very long. And he found this bacteria. Alpha Proctobacteria, Gamma Proctobacteria, Actinobacteria. So this is a really nice example of urban microbial research in creativity. Like looking at the microbes inside this box. And in this way, we describe new insects and new microbes from urban areas. So we don't have to go far away. Here, probably you have these insects here in your room. You have oh, you know about it. Yeah. So yeah, you can have microbial ecology here in your own city or your home place. So with this, I would like to finish, and I have these home take-home messages. So the power and impact of microbiomics are enormous. I just give you a brief introduction that I hope you will remember forever about your microbial partners. So don't ever feel lonely. You can do a lot of research questions. There is a lot of potential for students in Mexico, abroad, wherever. Microbes are always with us. And this is important for you. Well, I hope it's important for you. So don't grow fast, be young. That's important. Be young, be passionate about the things you like. You learn more from mistakes than from successes. So most of the time you fail and you receive no, sounds worse. But sometimes you receive a yes, and that's important for your life and your travels or whatever your opportunities. Please remember that everyone has their own path. Don't ever try to do the same thing as others. Don't try to compare with your friends or family members or whatever, because everyone has their own path. Enjoy your friends and your loved ones and travel the world. With that, I would like just to provide an acknowledgement for your university, for the organizing committee, and especially to Nicolas and Javier for the invitation. It's my first time in the university, so now so good so far. With these two societies that are with close friends. And with that, thank you very much for the attention.
Hey guys, you can ask questions. Remember, you can ask questions in English. If you feel comfortable in Spanish, they are also welcome. So, who has a question? Una pregunta? ¿Alguien?
Sí, de eso.
Porque tienes una variedad grande de microorganismos que usan diferentes estrategias de ensamblaje. ¿Es muy diferente o es la misma software?
No. Muy buena pregunta. Depende de dónde quieras empezar. Cuando tú sabes que tu comunidad incluye varias poblaciones de microorganismos, tienes que tener cuidado desde el momento en que sabes el DNA, porque generalmente se utilizan kits de extracción de DNA, pero muchas veces los kits no son el mundo ideal. Es muy práctico, pero lo ideal es hacer varias extracciones de DNA usando métodos que involucren temperatura, alguna cuestión mecánica o algún tipo de solvente químico para tratar de realizar la mayor cantidad de células. Hay bacterias que resisten muy bien los tratamientos de los kits. Si tú sabes que tienes una muestra de suelo que tiene una gran cantidad de microorganismos, un solo kit no te va a funcionar a la perfección. Después, tienes que elegir muy bien cuál va a ser tu estrategia para secuenciar. Digo, ahí vienen tres nombres: Illumina, PacBio y Genomic. Illumina produce una gran cantidad de lecturas cortas de muy buena calidad y son económicas. Entonces da mucha cobertura de lo que se secuencia de la comunidad. Mientras que, por ejemplo, PacBio te produce lecturas muy largas de calidad media a alta ya, pero son más costosas. Entonces, si tú sabes que tu comunidad es altamente diversa, tal vez lo que te conviene es tener mucha cobertura, y si el presupuesto es limitado, es mejor secuenciar con Illumina. Bueno, ya que tienes los datos, como los hayas tenido, viene la parte de análisis. Lo que siempre se tiene que hacer, incluso si ustedes utilizan algún servicio comercial con algún centro de secuenciación, es siempre revisar la calidad para ver que ahora sí que lo que pagaste. Sí. Dependiendo de eso, vas a tener un volumen de datos. Muchas veces el volumen de datos ya es tan alto que necesitas utilizar programas o en la nube o en un servidor de cómputo. Los programas que utilizas, los ensambladores, normalmente están especializados para comunidades muy simples o genomasterizados. Entonces, la mayoría de los programas, voy a mencionar uno, Space, ya tienen versiones como MetaSpace, que te permiten justo analizar los datos específicos para lo que estás preguntando. Entonces, si estás haciendo metagenómica, lo ideal es utilizar más de 2 programas para poder obtener ensambles del mismo set de datos y poder verificar que los métricos, o sea, pues no necesariamente van a coincidir, pero se van a implementar. En la actualidad, lo que se está haciendo más bien es tratar de recuperar los famosos maps, los metagenomas en los genos. Y para eso necesitas programas que se llaman en conjunto binding metagenómico. Entonces, ahí hay varios programas que intentan hacerlo de la mejor forma, pero a la actualidad sigue siendo bastante difícil en comunidades que son complejas. Lo mejor que puedes hacer es utilizar varios programas para contrastar esos bins. Entonces, ahí puedes darte cuenta muy rápido que en los resultados, unos programas funcionaban mejor para recuperar más de los organismos que de otros. Entonces, respondiendo muy puntualmente, los programas de la informática los puedes usar con cualquier set de datos. Sin embargo, va a haber algunos que son especializados para el tipo de preguntar, el tipo de datos que tú tienes o generas. Gracias. Sí. Sí, y luego su compañera.
Para los extremos que nos están analizando en los azufres, nada más están considerando temperatura y pH, o por ejemplo están tomando en cuenta factores de salmueras de preparatos, de presión.
Sí, bueno, cuando fui la primera vez en los azufres, primero tuve una entrevista con la gente que trabaja en el CPS, fueron nuestros primeros días. Y justo nos comentaban que para ellos una de las grandes dificultades de la generación de energía térmica es que como residuos se produce agua salmuera, que básicamente es un agua, no, es un agua muy saturada de sales, de muchos tipos, y muchos metales pesados. Entonces, esa agua que se produce la almacenan en la superficie, en pocitos al aire libre. Ya no lo tienes que pasar en su coche o hacer frío. Y entonces el tema es que parte de esa agua se llega a evaporar en la superficie y se empieza a acumular una sal que es muy saturada en metales pesados. El detalle es que hacen esto para que después en el proceso de generación de energía la reinyectan al pozo para que esto sea sostenible o lo más sostenible posible. Les vuelven a reinyectar este líquido para mantener pues, el acuífero. El detalle es que se acumulan estos sitios con microsal. Entonces, una de las preguntas que ellos nos hicieron en su momento es ¿qué microorganismos podrían estar viviendo en estos pocitos de salmuera para ver si nos pueden ayudar o no? Y la respuesta fue muy bonita, sí, porque tienen genes que justo lo que hacen es metabolizar estos compuestos tóxicos y sorprendentemente los convierten en otros que son menos tóxicos y que son más fáciles de eliminar del agua. Entonces, sí, o sea, no necesariamente todo es como temperatura, acidez, también estamos viendo las cuestiones de salud pública o de conservación de la biodiversidad, aunque pueda ayudar a los procesos, pues, en este caso, de producción de energía. ¿No? Sí.
Habías dicho que el 95% de las proteínas que producimos está en las instituciones. Entonces me gustaría saber más en cómo es que se produce, o si tiene algo que ver con la proteína que consumimos, con cómo está relacionada con nuestras emociones, ya sea si estamos tristes no comemos, o cómo tiene que ver eso con nuestras proteínas.
Qué buena pregunta. Pero me da miedo responder en menos de la parte de clase. En corto, muchos de los, o sea, hay muchos alimentos al momento que los digerimos o los que tienen los microorganismos dentro de nosotros, pueden proveer precursores de algunos neurotransmisores o incluso algunos de estas moléculas pueden servir ya como neurotransmisores. Y entonces, algunos pueden entrar por receptores en las células epiteliales y algunos otros directamente ya afectan al sistema nervioso. Entonces, ¿cómo se producen los neurotransmisores en estas vísceras? Pues puede ser por el mismo metabolismo de la parte humana, o la parte microbiana, o un complemento. ¿Cómo actúan? por difusión o por receptores especializados en las membranas de las células epiteliales, principalmente. Y luego la otra parte es cómo afectan las emociones. Bueno, es que generalmente cuando hay un desbalance en las comunidades microbianas, en cualquier parte del cuerpo, eso se va a traducir en un fenotipo. Algunos de estos fenotipos pueden ser correlacionados con algún síndrome o enfermedad. Entonces, no sé, si tal vez tú no estás generando suficientes neurotransmisores del que tú gustes, o que alguno de estos receptores no lo esté recibiendo, o no a la suficiente concentración, eso puede provocar algún tipo de emoción, o síndrome, o enfermedad, dependiendo de qué se trata. Entonces, por eso es importante tratar de llevar una vida equilibrada. Se entiende por ser equilibrados. Pero particularmente la dieta se sabe muy bien que puede estar relacionada con ciertos fenotipos relacionados a salud o a la enfermedad. Las dietas mediterráneas siempre han sido asociadas a un estilo de vida saludable, no sé, un buen índice de masa corporal, una longevidad bastante notoria respecto a otras zonas geográficas. Y lo que se ha visto es que hay correlaciones con los microorganismos que están presentes en los intestinos. Entonces, es un trabajo de comunidad. Entonces, son preguntas complejas, son temas complejos que tienen multicausa. O sea, no solo en la parte genética, no solo en la parte microbiana, o sea, incluye mucho tu genética, incluye mucho el estilo de vida, influye mucho qué estilo de vida llevas, porque, bueno, eso no lo comenté, pero no es lo mismo llevar un estilo de vida en una tribu en el Amazonas o en cualquier lugar del mundo versus nosotros que vivimos en la ciudad. Entonces eso cambia mucho las comunidades que tenemos asociadas. Les pongo un ejemplo clarísimo. ¿Qué hacemos para el que se baña en la mañana o en algún momento del día? A la gran mayoría de nosotros nos gusta el agua caliente. Imaginen sus microbios en la piel. ¿Cuántos microbios? Caliente, hirviendo. De hecho, había mucho jabón, un detergente químico en la piel. Y luego, ¿qué crees? Me pongo la crema carísima y me pongo loción. Me lavé los dientes. Estoy acabando con biofilms de la boca. Me estoy vistiendo con fibras sintéticas que pueden cambiar la composición de mis microbios en el cuerpo. Hay muchos factores. El estilo de vida, qué comes, con quién sales, cosas curiosas y rápidas. Cuando nacemos no tenemos las bacterias que nos provocan las caries. Esas las adquirimos. ¿Tienes al bebé? ¡Ay, qué bonito bebé! Estás hablando, ahí van partículas de saliva. Y esa es una forma de inocular a los bebés. Si tuvieron la gran suerte de que llevaban ciertas cepas de estafilococos, pueden provocar caries. Algunos tipos de cáncer oral, garganta, no tienen nada que ver con tomar o fumar, sino con las personas con las que tuviste relaciones sexuales. Son microbios, son virus, son los alertas, que se transmiten de esa forma. Y eso lo sabemos por varias campañas de no fumes y no tomes en Estados Unidos, que de todas formas veían que ciertos tipos de cáncer seguían incrementándose. Y se dieron cuenta que no tiene nada que ver con fumar o tomar, eran otro tipo de medidas que se deberían tomar. Entonces, todo eso afecta. Entonces, no es una respuesta pues, sencilla de dar, porque en realidad es multifactorial. Es lo que mejor te podría decir. Listo.
Como soy secretario, una pregunta muy rápida. En tu opinión, ¿qué va a pasar con los virus? ¿Van a seguir siendo los seres vivos o se los van a reclasificar después? ¿O qué va a pasar con estos?
No, otra clase. No, fascinante. En pocas palabras, fíjate que eso a mí también me encanta. Me movió mucho una vez que fui a un congreso, conocí a un investigador que se llama Patrick Porter. Él los ve como máquinas virales. Usa un ejemplo, digo, seríamos una imagen. Imagínense una neurona. Si yo les pongo una imagen de una neurona, ¿me dirían que eso es un humano? Pues no. Pero si yo les pongo una partícula viral, ¿me dirían que eso es un virus? De acuerdo a lo que propone Patrick Porter, es no. Esa es la forma inactiva de un virus. ¿Cuándo es activo el virus? Cuando infecta a su célula puesta. Está hackeando literalmente el metabolismo de esa célula. Esa célula estaba programada para hacer metabolismo, pero ahora hace lo que el virus le dice, que básicamente es genera nuevas partículas virales, lisa la célula y viene a infectar nuevas, para tener más partículas virales. Entonces esa célula ya no está haciendo su propio metabolismo, está haciendo lo que el DNA del virus le mandó a hacer. Y entonces, para diferenciarlo, lo propone un término que son las fábricas virales, de viral factories. Entonces, lo utiliza para referirse en ese momento a la parte sí activa de un virus. Entonces, una partícula viral es la parte inactiva, el viral factory es la parte activa de un virus. Y en ese momento podríamos pensar que un virus está vivo, porque está haciendo el metabolismo de la célula. No es el metabolismo de la célula original. Entonces a mí me da miedo cada vez que me da una gripita, ¿no? Porque eso de tener ahí una cosa viral haciendo su propio metabolismo como que da un poco de pánico. Definitivamente es una pregunta abierta. Lo que sí sabemos es que los virus no se les considera como vivos por el hecho de que no pueden sintetizar sus propias proteínas. Si lo vemos así, pues eso está, digamos, primaria, ¿no? O sea, la vida necesita tener un metabolismo propio. Los virus, la mayoría de ellos no lo puede tener de forma independiente. Si lo vemos así de tajante, pues están muertos. Pero si vemos estas otras visiones más modernas, pues sí podría haber ciertos momentos o estados emergentes en donde podríamos decir que el virus está vivo, pero no refiriéndose como virus, sino como fábricas virales. Sí.
Ok, thank you very much. Muchas gracias. Tenemos que continuar. Muchas gracias. Ah, sí,